All right, for Introduction to Psychology, this is Biological Psychology, Chapter 3. Right, so our objectives for this particular chapter is to be able to identify the main structures of a neuron, be able to describe what an action potential is, talk about the state of all or none law of an action potential. Also, we'll describe communication and synapses, and then we'll start opening up and speaking about how drugs affect behavior or changing actions within a synapse. That's probably going to be the extent of this first video lecture. However, in terms of your reading, you should continue forward because you'll also want to be able to distinguish among the different classes of drugs, such as stimulants, hallucinogens, anoxacylics, as well as opiates. Hopefully, the chapter will be able to explain to you how a drug may affect one immediately as well as at different times or different effects hours later. We'll talk about how scientists favor the position called monism. I'll also have within our discussions citing examples about brain damage and how that will impact behavior. We'll also take a look at the different methods of how we might measure brain activity. By the end of the chapter, you will be able to cite what brain plasticity is and give me some examples. Also, we'll talk about binding and the problems with that there. Talk about the basic principles of genetics and discuss epigenetics as terms of a mechanism that would help to influence one's development. Then we'll go through how researchers looks at heritability and then initially describe the assumptions and the goals of evolutionary psychology. But as I mentioned, we're probably going to focus more on the first slide of objectives within this video lecture and move forward on it again, either in another video lecture or in class face to face. So let's get started by taking a look at neurons and behavior.
Now, I'm sure this is a refresher for most of you, but let's take a little journey through the life of a neuron. Imagine you're at a party. Don't worry, there's pizza. And your job is to pass along juicy bits of gossip. That's pretty much what neurons do, except the gossip is information, and they do it using electricity and chemicals. Our starting point is the dendrites. Think of dendrites as the neuron's many little ears or tree branches. They're sitting there eagerly waiting to hear the gossip, aka signals from neighboring neurons. Dendrites pick up incoming messages and send them toward the main hub of the neuron. Now, the next stop would be the cell body or the soma. The soma is like the neuron's main office. It has the nucleus, the boss, and handles all the cell's day-to-day operations. It also decides, is this gossip juicy enough to pass along? If the signals collected by the dendrites are strong enough, the soma says, all right, let's spread the word. The long road, the axon. Once the soma gives the green light, the signal travels down the axon. Picture the axon as a long hallway or even a telephone wire. Some axons are covered in a fatty coating called the myelin sheath, like insulation on a wire, which makes the signal travel much faster. Without it, the message might just crawl along, and no one likes slow gossip. Time to look at the pit stops, the nodes of Ranvier. Along the axon are tiny gaps in the myelin sheath called nodes of Ranvier. These act like turbo boosters, letting the signal jump down the axon more quickly. It's the neuron's way of using express shipping instead of snail mail. The grand finale terminal buttons, also known as axon terminals. At the end of the axon are the terminal buttons. This is where the neuron sends out its gossip to the next crowd, but instead of just yelling it across the room, it releases chemical messengers called neurotransmitters into the synapse, the tiny space between neurons. The next neurons, dendrites, catch those neurotransmitters and the cycle starts all over again. Boom. Gossip successfully delivered. Here's an analogy to help put it all together. Dendrites listen, soma decides, axon carries, and the terminal buttons share. And that's the neuron's life in a nutshell. It's like the ultimate party network, except instead of drama, it's passing along the signals that let you think, move, feel, and yes, even decide which pizza topping you like best.
All right, so I want to discuss the communication styles of neurons. More or less, a neuron's chatting occurs through resting potentials, depolarization, as well as action potentials. I want you to think about your neurons being these tiny social butterflies. And more or less, what happens is gossip. You know how people love to gossip. Well, the neurons, they like to gossip with each other too. However, instead of whispering over lattes, what they're doing is sending these electrical signals out. And here's how a conversation might work. It'll start with a resting potential, whereas the neurons in that chill mode, and then it'll start talking. And it's chilling is what's called that resting potential. So more or less, think of it as when the neurons in the default mood, woosah, calm and ready. However, on the inside, the neuron is more negatively charged and compared to the outside. Now that really happens because the ions, which are these tiny charged particles, like sodium as well as potassium, um, and it's those special ion pumps that'll keep the sodium mostly outside and keep the potassium mostly inside to help maintain the charge. That's the difference there. And more or less, it's like the neuron would be holding back, waiting for something exciting to happen. So, although I don't have it written here, you should know what a depolarization is. This is how that excitement starts to build. And you can imagine your neuron is starting to hear that juicy gossip. And it's also known as a signal from another neuron. But it's exciting enough that it's going to get those sodium channels to open up, and then the sodium ions will start rushing inside that neuron. And whoa, suddenly inside the neuron it's going to become less negative and even briefly positive. It'll be this change that we call depolarization. It's like the neuron's going, wait, what? Tell me more. Now, for what you see on the slide is the term action potential. This is what we call that big message. So if the depolarization reaches a certain threshold, like hitting the neuron's hype limit, then what happens is it'll trigger an action potential. And that's going to be that rapid electrical signal that'll zip down the neuron's axon like a wave at a football game. Now, more sodium will flood in, and then that potassium is going to start to exit out in order to reset the charge. And it'll be that signal that moves right along. This allows the message to travel fast and reach the terminal buttons at the end of the neuron. So what happens after somebody gossips? Well, it's like you get reset. And what happens after an action potential passes is more or less when the neuron is going to return to its resting state. Back to chill mode. So it's ready for that next message. Potassium and sodium pumps work like the neuron's cleanup crew, and it's going to restore that resting potential. So more or less, when you think about a resting potential, that's when the neuron is being ready but quiet. It's going to be negative on the inside. Just remember what I said about depolarization, because that's necessary, and that's when the sodium rushes in, the neuron's going to get excited, leading to the action potential. And that's when the neuron shouts that message down that axon. Then what happens is we reset and wait for the next message to come about. Now, one other thing I want to mention is the all or nothing law, or all or none law, which states more or less that an axon cannot vary the strength or velocity of its action potentials. It's either going to do it or it's not, plain and simple. So as I mentioned in the previous slide, what happens with the action potential is when the sodium gates, and I didn't call it gates before, but let's call it the gates now. They're going to open up and the sodium ions will enter the axon bearing a positive charge. Once that action potential occurs, the sodium gates will then close at the point and open up at the next point to allow the axon to move things forward. Once that gate closes for the sodium, potassium gates open up, and that's when the potassium ions flow out of the axon. So I wanted to give you the more scientific terminology of that process, rather than talking about it simply as gossip, because I want you to make sure you're understanding that reading. However, if you do have a question, do not hesitate to ask. Now it's time for me to trip you up a little bit, because within the discussion, I might have presented some things to you, but you tell me what you're seeing, okay? Which of the following helps to synchronize activity among neurons and assist in formation processing? So, one, would that be myelin? Or could it be dendrites? Might it be axon? Or could it be the glia? What do you think? All right. Now, I didn't talk about this term to you in the very beginning, but I did present it to you on a slide when we're talking about glia cells. Now, the glia really is helping with the myelin sheath in that it insulates the neurons, it synchronizes activity among the neighboring neurons. It'll help to also remove waste from your neurons, as well as assist in information processing. It's going to get things moving along. That myelin sheath that I mentioned, that's how everything gets pushed, okay? So neurons, though, I want you to know, outnumbers the glia in some brain areas, but then there's more glia in a lot of other areas of the brain, okay? All right, what is a synapse? I want you to think of the synapse as this tiny meeting point between two neurons, where they'll pass messages back and forth through one another. However, here's the twist to this. Neurons don't actually touch. So realize there's a microscopic gap that's called the synaptic cleft. And it's here that it'll help to work those communication skills between neurons out. Looking at this step by step, we talked about the axon potential. So it's going to start with the axon potential. It arrives, bringing that electrical signal that we talked about, allowing it to travel down the axon, and is sending to the neuron, or in this case, understand it as the presynaptic neuron, and allows it to reach the axon or axon terminal. Then what happens usually is the neurotransmitters will release, and know that that's a chemical that activates receptors of other neurons, okay? this happens with these little sacs called vesicles, and it's going to release the chemical messengers or the neurotransmitters into the synaptic cleft. Again, that tiny gap between neurons. The next thing that happens is it'll start crossing the gap. Neurotransmitters will start to float across a gap, and much like paper airplanes flying to the next neuron, you'll then have the message received. And that's going to be on the receiving neuron or the postsynaptic neuron. There you'll find where the receptor sites, they're going to be shaped like locks so they can grab them and hold them. The neurotransmitters will fit into those locks, just like if you were putting in a key. So once that occurs, it's going to start delivering the message. Once that's completed, again, we have reset, where after it's been delivering the message, neurotransmitters will either take back the message, or it'll send it on its way, or reuptake, or another way of thinking about it is broken down by enzymes to kind of help clear the space for the next message. If you're wondering why the synapse is important, well, if we didn't have synapses, the neurons will not communicate. It's necessary. Every thought, every memory, every movement, every feeling, basically everything your brain does relies on our synapses working properly. So that, in a nutshell, tells you what synapses are. I know I got a bad habit of repeating myself. You might hear me say nutshell in another 100 different terms. However, when you look at the whole picture, there's a new term. That's what you want to see. You want to know about presynaptic endings, neurotransmitters, and postsynaptic neurons. This is kind of an image of what I just described on the previous slide, so I'm going to just tell you to take it in, look at it, and learn it. Oh, here I go again, throwing in questions in regards to information I might not have shared. Now, why do you think I do that? Because I want you to make sure you're reading the chapter is why. Understand that when I give a lecture, I'm going to give you the bulk of information. However, it's expected that you will be doing your reading. And you will find this information in your textbook. So please make sure you're reading a chapter. And do not anticipate or expect me to give you word for word account of what's in your textbook. That's your job to gather. I'm only going to help by giving scenarios, analogies, examples of the material that you should be reading about already in order to help you to understand what it is. With that being said, here's your next question. Which of the following diffuse to a wider brain area to produce effects that last minutes instead of seconds? Would that be peptide transmitters or inhibitory messages? What do you think? Do 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 do. All right, so the answer would be peptide transmitters. And that really depends on the transmitter we're talking about as well as its receptor. And that's because it can or might have a sudden onset that's milliseconds long, not even a whole second. And it might develop gradually and last for several seconds. When we're talking about peptide transmitters, know that it's going to help to diffuse the wider brain area that'll help produce effects that last for minutes instead of just milliseconds. So that means a quick, sudden message that can be deemed important is like when we see something in our vision, or if we hear something and our brain says, oh, gotta get this now. Gotta grab what I was able to gather. Slower, longer lasting messages they're going to be more appropriate when it comes to when we're talking about taste or smell. And they're locked more in your memory much quicker for long-term memory. And that's because of semantics. And we'll talk later down the line about that in sensation and perception. But for now, we'll stop. Just want to give you a little understanding about those peptide transmitters.
So now let's look at this in an everyday scenario. Imagine your friends are trying to coordinate a pizza night. Okay, what can I say? I love pizza. Anyway, this is how it would probably go. Starting with the neurotransmitters, the messages. Your phone sends a text, pizza at 7? This is like a neurotransmitter. The actual message leaving your phone, sending neuron, and traveling to your friends, receiving neurons. The receptor sites, this is who gets the message. Your friends' phones have the right app, the lock, to receive your text. If the app matches the format of your text, it opens it. That's how receptor sites work. Only the right key, message, fits the right lock, receptor. Reuptake, message recall. After you send the text, your phone still has a copy in the sent folder. That's like reuptake. The neuron pulls some neurotransmitters back in to reuse later. No need to keep spamming new messages when you can recycle. So now comes the enzymes. This is the cleanup crew. If someone accidentally sends 10 pizza emojis after the conversation's done, you delete them so the chat isn't clogged. Enzymes do the same. They break down extra neurotransmitters left floating in the synaptic gap so things stay organized. Then the neuromodulators, considered the volume control. Let's say your group chat is on Do Not Disturb for one friend, while another has it pinned and gets special alerts. That's like neuromodulators. They don't send a direct message, but adjust how big of an impact the signals have. Some make neurons more alert, others calm things down. Therefore, your brain is basically one giant super fast group chat where billions of messages fly every second. Without neurotransmitters, receptor sites, reuptake, enzymes, and neuromodulators, all that communication would be chaos, like trying to plan pizza night in a 500-person group text with no rules.
All right, we're ending it here. Um, as you can see, I have a QR code there that I'll need you to scan so that your attendance can be taken. I will make sure to say in the announcement that you do have to review this lecture by 11:59 tonight. Um, I do apologize for making you go after class time. Um, However, it's important because you need to be able to see and know that there's a QR code that will need your scanning so that I can uh, have your attendance for the day. It was going to either be that or give you another additional written assignment, which I didn't want to do that because you already have an assignment that's due Friday. So instead of that, just make sure that you're watching and listening at the video lecture, and this is your attendance for the day. So, with that being said, again, thank you for your patience. I do apologize. I'm going to let you know already. Um, this whole week is I won't be back, and hopefully I'll be able to return back to campus by the well, we have Labor Day Monday, so that next Wednesday I'm hoping to be back in full swing of things, and that we get to see each other and converse and have face to face again. I'm slowly but surely getting on the mend. I hope to be at 100% by next Wednesday. But with that said, this Friday You will receive another video lecture, and it's going to be continuing chapter 3. With that said, I thank you so much for listening. I again apologize for all the inconvenience that you've been put through, and just stay safe and stay healthy for me, okay? With that said, have a good one. Thanks for listening.